All right, welcome to lab number 3. This is the lab on dilutions. So if you read the chapter lecture, the PowerPoint slides, you saw that it actually went through how to make a dilution. And this was taken directly from MLT 115 class laboratory calculations for chapter 3. And when you look at your handout for the lab today, you will see that it's telling you to set up tubes and use a pipette and make some dilutions, which obviously we're not going to do in person, but I'm going to do that for you here. So part of that lab handout is to determine the volume of colored solution. I have red colored solution here that is going to need to be used in order to make each dilution, and the amount of diluent, which is going to be water today, that needs to be added to dilute the red solution. If you have not performed those calculations, or the calculations at the bottom that ask you for the concentration of each of the tubes, stop this video and perform those calculations now, and then come back to the video. If you have already done the math, however, what I'm going to do is I'm going to go through each of these one by one and make those dilutions with you. So the first practice dilution, number 1, said to make 3 tubes, and each of the tubes is going to be independently made, or an independent dilution, and each is going to be a 1 to 10. So I'm taking 3 tubes and I'm labeling them 1 to 10 for the dilution I'm going to make. And I have calculated that I need 100 microliters of the red solution in order to make this dilution a 1 to 10, and I'm going to need 900 microliters of water diluent added to that red solution in order to have a total volume of 1 milliliter, which is what is requested for this problem. So I'm setting my adjustable micropipetters to the proper volumes. The first thing I'm going to add to each tube is 100 microliters of red solution. I am going to use the same pipette tip for this because it's simply the same red solution being added to a clean tube. Next, I'm going to take my microliter pipetter at 900 microliters and add the water. If you remember from phase 1, whenever you are aspirating the proper volume, you push the depressor to the first stop. You fill your tip with the solution, and then when adding it to the new container that you're transferring the liquid to, you push to the second stop to make sure the full microliter amount is dispensed. And so now I have 3 1 to 10 dilutions. Okay. Number 2, or the second dilution practice, is asking you to make 3 independent dilutions, but these dilutions are not all the same. The first is a 1 to 2 dilution, the second is going to be a 1 to 5 dilution, and the third is going to be a 1 to 10 dilution. So when I did the math for my 1 to 2, I found out that I was going to need 500 microliters of red solution, and since I need a total volume of 1 milliliter, I need to add another 500 microliters of water. For the 1 to 5 dilution, I'm going to need 200 microliters of the red solution, and in order to get a total volume of 1 milliliter, I am going to add 800 microliters of the water. And for the 1 to 10, we did that calculation for the previous example, and it is 100 microliters of red solution and 900 microliters of the water diluent. So my 1 to 2 has 500. I'm getting a new tip for my water, and 500 to make 1 milliliter. I'm now setting my diluent pipetter to the 800 and my red solution pipetter to 200. And for my last tube, 100 microliters of red solution and 900 microliters of water. I'm going to mix each, and as you can see, the 1 to 2 dilution is much darker than the 1 to 5, and the 1 to 5 is much darker than the 1 to 10. The next example in the lab is going to be a dilution series. So if you remember from MLT 115, a dilution series or a serial dilution is a group of tubes, and each of these tubes has the same dilution in it. The difference between a serial dilution and those independent dilutions we just did is that we're using some of the dilution from tube 1 to make tube 2, some of the dilution from tube 2 to make tube 3, and so on and so forth. So I have pulled out 5 tubes and labeled them 1 through 5. The dilution I'm supposed to be making in the first serial dilution is a 1 to 2 or a 2-fold serial dilution. So my calculations for making a 1 to 2 with a total volume of 1 milliliter would mean I need to use 500 microliters of red solution in each tube and 500 microliters of water diluent. The great thing about serial dilutions is because every tube has the same amount of diluent in it, you can add the diluent to all of your tubes before actually adding the red solution to be diluted. So I'm simply adding 500 microliters of water diluent to each of my tubes. And I can do that with the same pipette tip because all I'm doing is transferring water to clean tubes. Since I also need 500 microliters of red solution to make the 1 to 2 dilution in tube 1, I then add my 500 microliters and I mix using the pipette tip. I am mixing by simply depressing up and down. After I feel that the solution is adequately mixed, what I'm going to do is press to the first stop on my micropipetter and remove the amount that I need to make the next dilution in tube 2, which is 500 microliters. I'm transferring that to tube 2, which also already has 500 microliters of water, and I'm mixing. Then I'm removing 500 microliters and moving it to tube 3, and I'm mixing. And when I'm mixing, I am keeping the tip of the pipette in the liquid so that I don't create bubbles in my dilution. Transferring to tube 4, mixing, and lastly transferring 500 to tube 5 and mixing. So you can see that it was much quicker to already have my water diluent in the tubes so that all I had to do was move the red liquid from tube to tube after making the previous dilution. You can also see that as you go from tube 1 to 2 and so on, the solution gets much lighter because I continued to dilute and redilute and redilute that solution that was added to the first tube. Make sure that you do determine the concentration of each of these tubes at the bottom of page 3 in your lab handout. The next serial dilution also contains 5 tubes, and this dilution is going to be a 5-fold. 5-fold means it is a 1 to 5 dilution in each of the tubes. We are still going to be making 5 tubes, so I'm labeling tubes 1 through 5 now. And when doing the math for a 1 to 5, we find out that we need 200 microliters of red solution to make a 1 to 5 dilution in tube 1. And then we need, in order to have a total 200 microliters of red solution with a total of 100 milliliter, would be 800 microliters of water diluent. Because every tube is going to have a 1 to 5 dilution, because it's a 5-fold, we can go ahead and add our 800 microliters of water to every tube in this dilution series. When I'm pipetting, I'm also making sure that I am touching the tip of my pipette to the edge of my tube so that my fluid that I'm adding is running all the way to the bottom of the tube and not having little drops stuck at the top of the tube, because we want to make sure that whole 800 microliters gets mixed in the end. Okay, our 200 microliters of red solution can now be added to the first tube to make the 1 to 5 dilution. I'm pipetting, mixing using the pipette by depressing up and down with my thumb. After I feel it's thoroughly mixed, I am removing 200 microliters from tube 1 and moving it to tube 2. Again, mixing. I'm keeping the pipette tip in the liquid to prevent bubbles. Moving to tube 3 and continuing on all the way through tube 5. You will notice that this 1 to 5 dilution gets much lighter much more quickly than our 1 to 2 dilution did. So we have a much larger difference in the concentration as we move from tube to tube. Don't forget to calculate those concentrations in each tube for tubes 1 through 5. All right, in serology, the whole reason that we are actually performing this lab is because in serology we make serial dilutions all the time. When we make a serial dilution of a patient's serum sample in the serology lab, we're trying to find out just how much of something is in their serum that will cause a reaction with our testing kits. And our testing kits all contain cards, and the cards have reaction circles on them. You have a picture of a card with reaction circles very similar to this in your lab handout. What we will do is we will simply consider this reaction card to be like the tubes that we just make dilutions in. This could be tube 1, 2, 3, 4, 5, and so on. So there are 10 spaces to make dilutions on this card. The reason we would do this is if a person had a very high amount of a certain antibody in their sample, we may need to dilute that sample down in order to see an adequate reaction in a serology test. So what I'm going to be doing is making a 1 to 2 serial dilution on this card for you today. Whenever we are using these reaction cards, we don't use as high of a volume of sample or diluent as we do in the tubes, because there's only so much space for us to see a mixture of two things happening in these small circle reaction spaces. So usually our volume that we'll use is a 40 microliter total volume. So if I am going to be making a total volume of 40 microliters and I'm making a 1 to 2 dilution, I'm going to need 20 microliters of red solution and 20 microliters of water. So I have set my pipette to 20 microliters. And because I know that each of these reaction circles, just like each of my tubes I made in my dilution series previously, is going to have the same dilution, a 1 to 2, I know that each of these reaction circles needs 20 microliters of diluent or water. So what I'm going to be doing is placing 20 microliters of water onto the reaction circles. Okay, and what I'm going to do is I'm actually going to try to get a close-up for you so that you can see what this really does look like on the card. These cards are covered with a wax, which allows that liquid to remain as a small droplet on the card. Okay, and so as we're doing this, that water, that diluent, is not actually going to go anywhere. It is going to remain on the card itself. Okay. And so now all I need to do is I need to add my sample, my red solution that is going to be diluted. Okay, again, I'm going to be using 20 microliters. So what I'm going to do is I'm going to take 20 microliters of red solution, add it to the 20 microliters in circle 1, just like I would if it were a tube. And I'm going to mix using the pipette tip until it's mixed thoroughly. And then I'm going to move 20 microliters from circle 1 to circle 2. Mix. Move to circle 3 and mix. Circle 4. Circle 5. And so on. So now after I would make a dilution of patient sample using this serial dilution procedure, the way I would test it is I would take a reagent from a serology test, mix it with the serum, and get results as to whether or not a certain analyte was present that we're testing for. So as you can see, we get a change in color as we go from a higher concentration to a lower concentration. And what it's asking you to do in your lab handout is actually calculate those concentrations. Circle 1 has a 1 to 2. Then we're taking some of that to make another 1 to 2 in circle 2. So that's a 1 to 4, and so on. So you need to write the concentrations for each of the circles in the lab handout on the last question. As always, if you have questions, please email me and make the title of your email the lab dilution video. Thank you.